Hello, brave biology class. Welcome to the fungus labs. So, we got to complete all of our fungus labs today. We missed you guys who were absent. We had a great time. We did not end up with pumpkin or squash mold, but we did end up with bread mold and cucumber mold. And this is what our mold looks like in our textbook. With the labels of the sporangium, the hypha, and the rhizoids. And this is what our textbook mold sporangium looks like with the little spores. What we saw looked more like this. So here's the bread mold at medium and the bread mold. These were two different slides of bread mold at high power. And here's the cucumber mold on high power. This was one of what one of our cucumber molds looked like. It was pretty cool. It was kind of spread out. And then the other one was a little thicker, so you could see the strands. It was you could see a little bit of the spores, but you saw a lot of strands as well. It was very exciting. So that's, oops, sorry about that. That was page 76. Now on page 77, it asks you to fill in the diagram. So your rhizoids are down here in your fungus. These are your stolons connecting, your hypha, your spores popping out of the sporangium here. And here's some little babies right here. See? Page 77. Those were really cool. We also got a mushroom. We actually had two mushrooms. Now we didn't quite get, here's the outside of the mushroom with the cap, the gills, and the stipe. We didn't get this part. It was broken off here, but it was still a good mushroom. And then we did the, what did I do? The scrape method on the bottom of the gills to come up with the spores from the basidia. And that was medium. That was like medium. And that was pretty easy to see. It looked just pretty much round. The question, if you do a slice, why must it be thin? Oh, you can't see that. If you do a slice on number 3, slice prep, why must it be thin? We didn't do a slice, but it must be thin because if it's too thick, the light can't see through, and then you can't see what's going on in the specimen. The next page is page 81, and it has your picture of your mushroom here, your drawing of your mushroom, and they just wanted you to label where the different things are. The basidia is here, which they're made to look like clubs here, and that's where your spores are. Your caps on the top, your gills, of course, are your little fanny things on the bottom here. Your stipe and your mycelium is, of course, at the bottom. Isn't it interesting that all this is just the reproductive part of the mushroom, of the fungus, and this is really the plant, or it's not really a plant or an animal, but that's really the fungus part of the mushroom. I think that's really cool. Number 5, on what structures are the spores of the mushroom found? And the spores are housed in the gills of a mushroom, as we saw with this specimen. And we also got through the yeast lab on page 84. Here's the picture of the yeast. The yeast pretty much looked like little baby circles. We tried to find them budding, which is really with the little pieces coming off and then separating. We didn't see a lot of that, but we did see some active yeast. Number 2, what is the purpose of the sugar in the yeast and the purpose of the water? The sugar is the yeast food, and the warm water gives your warm environment for rapid growth and reproduction. Does yeast work if it's cold water? Yes, it does, but it doesn't work as fast. Does the yeast grow sexually or asexually? Yeast reproduces asexually by budding. Do you remember the mushroom can do, other types of fungus can sometimes do both. Explain the process of budding in the yeast. The budding is the new little circle. You got your little circle of yeast, and that starts to form another little bubble, and it gets bigger and bigger until it separates. from the new parent, from the new organism, and then it's a daughter cell. Number 5, what is the purpose and process of using yeast in baking? And I'll just read you what they have here. Yeast is used as a leavening agent in bread to make it rise. As the yeast feeds on the sugar, it releases carbon dioxide gas that's trapped as bubbles in the dough, and the bubbles give the bread its airy texture. So it releases carbon dioxide as it feeds, and it makes bubbles. Oh, and that's the end. So we did all 3 of those labs. Wow, there's not a lot of questions in there. We got through that very quickly. All right. And looking forward to seeing you on Monday.